live webinar where we are asking the question, how will CRISPR affect your farm? My name is Alex Martin, and I serve as a consultant to American Seed, and today I'm happy to be your host. As we go along, we invite you to share your thoughts and insight on social media using the hashtag ASWebinar. I also wanted to let you know that this webinar is being recorded and will be made available on american-seed.com following the event. During the presentation, you will likely have some questions for our speakers. I just ask that you simply type these in the chat box and we will address them during our Q&A session. Today's featured speakers are Nat Graham from the University of Minnesota and Michiel van Lookeren Campagne, representing Syngenta. During our time here, they will discuss what exactly is CRISPR-Cas9, how is genome editing different from creating GMOs, why should a farmer care about CRISPR-Cas9, what are some of the latest advancements made in agriculture for CRISPR, where the future of plant breeding will go with CRISPR-Cas9 capabilities, and then we will move into our Q&A session. First speaker of today is Nat Graham, a postdoctoral associate in the Voytas lab at the Center for Genome Engineering at the University of Minnesota. He received his bachelor's in science in biology from Truman State University and his doctorate in biology from the University of Missouri. Nat, thanks for being with us today to talk about CRISPR and the effects it will have on our farmers. I'll hand it over to you now.
Okay, so yeah, it's my pleasure to be here. Thanks for having me. So the questions I was kind of given as a primer to this, you just heard. And so it's kind of an interesting webinar we have today where I'll be giving the perspective from an academic lab background. So we're kind of on the ground floor doing research from the academic sector. And then later you'll hear from the private sector. So I think it'll be interesting to see how our two sides kind of mix together. So I think I'll just get right into it. So the first questions I kind of lumped together. So what exactly is CRISPR-Cas9 and how is it different from creating what traditionally are considered GMOs? And so from an academic perspective, I like to think about the organisms that we work with kind of as machines. So you can think of a plant that might make ethanol, for instance, or might make a pharmaceutical product. Those are kind of machines that are producing that product. And so you may have heard before that all these kind of machines and organisms, everything is run on a code called DNA. You've probably seen it look like this, where you have kind of a ladder, and the ladder is where the code is. And so organisms will read that code and then turn it into a product. So it works just like a machine would. So there's a little machine that runs through, reads the code, and spits out a product. And so what we're focused on from a genome engineering standpoint is how is it that we can manipulate this code to better function to better get products from what we're working with. So from an academic perspective, part of my job is kind of explaining how things work. And so we're kind of going to go into a little bit just to give people a better perspective how this works. So when we look at a DNA code, this is kind of what it looks like. It's just a bunch of letters, and it's not necessarily all that helpful from that perspective, but this is what we have to work with. And so from a science perspective, this is what we're dealing with. And so I kind of made an example that might make a little bit more sense to you, where I just use letters. So you can see there's a bunch of letters all together in kind of a paragraph. And so the first thing I understand about the DNA code is you can kind of break it into triplets. And so if you do that with our example, you can see these actually make words. You can see various words in there I made together. And additionally, when you start looking at the DNA code, you can realize that the words will sometimes string together into sentences. So something like this, where you have some strange sentences I made up where like, how far did you run is actually a sentence out of all these words. And so this is kind of how it works when we start looking at the genetic code as well in an organism. So we split it into triplicate codes, and then we realize when they all come together, they make a sentence, which is the product that's going to be made. So from a genetic engineer perspective, we're kind of concerned with, can we manipulate this code to make it say new things? Can you make it say what you want, or can you make it stop saying something it's already saying? So traditionally, with the GMO transgenics you might look at before, if we wanted to insert the sentence, the rat was fat, into this paragraph, how you would do it is you would just basically take that sentence and then inserting it into the paragraph, it would just kind of randomly insert itself. And so as you go through, you insert your new sentence, and then you find it somewhere in your paragraph. So you can see here, in blue, our new sentence is inserted. However, because it's inserted randomly, it's somehow disrupted the rest of our paragraph. So after our new insertion, nothing makes sense anymore, which is a problem. And so this is kind of how it works traditionally. You insert your new genetic sequence, and you have to go back and find it and see if it's disrupted anything. And so if it's disrupted something, you try again, and you try again, and you try again, until you find an insertion that's not disruptive. So in this situation, all the sentences behind it now make sense. And so this is the kind of insertion that we would be dealing with, where it's not going to cause a problem, so we would go forward and turn this into a product down the line. So that's kind of how it works with traditional transgenics. Okay, so the way it works in reality is you use either a bacterium or you use high-velocity particles to insert the genomic sequence, and it kind of randomly goes into the genome. But because of that, you can use pretty much any sequence that you want, insert it into the genome. And so while this might cause insertions into undesirable locations, it's generally okay because you can go through and find new sequences or new insertion points and go forward and make a product. If you wanted to turn something off, though, you're back to the traditional mutagenesis techniques. And those have been used for pretty much decades at this point, where you would use things like UV or X-ray radiation and radioactivity in some points. And that generally will mutate the genome, and then hopefully you can screen through and find the desired product that the plant is making you don't want it to make anymore, and you'll have it turned off. So as an overview, you are able to insert entirely new sequences, but you're also doing it randomly, so you have entire screening regimes, and it's not very fun to do. I've been on the other end of it, and you're doing a lot of characterization. So pretty much it's a really high-cost, low-efficiency process. So it's easy to do as far as inserting new sequences, but not necessarily turning off one. And so when we look at this compared to the new CRISPR-Cas9 technology, the bottom 2 points here are the most important, where you can insert sequences that are not native to the species with traditional transgenics, but you're also not reliant on the genome that you're working with. Now, if we compare that to what we're talking about with CRISPR-Cas9, this is the new tool everyone's very excited about. So this allows us to go through the genome, read through, and precisely alter the genome exactly where we want it to. For the most part, this is used to turn off sequences through mutation. Part of the lab that I'm in now, what we're working on is how do you insert new sequences. But for the most part, especially in the foreseeable future, a lot of the products will be coming from this technology or turning things off, making it produce less of something downstream. The way it works is it's kind of from an old bacterial system. So researchers figured out that bacteria have a defense mechanism. And the defense mechanism allows it to recognize sequences that are coming into it doesn't want. And if it recognizes those sequences, the Cas9 protein would cut them up. And so what we figured out was by slightly altering the machinery that bacteria were using, we could use it to recognize sequences that we tell it, and it would be able then to mutate those sequences just at one specific site. And so we kind of just co-opted a bacterial defense mechanism and are turning it into a product from that. So the way it one thing to note is that this technology is pretty new. From an academic perspective, it is really new. Really, 2012, 2013 was when it really started hitting the science journals, and that is not very long ago. And so this has been shown to work in a lot of plants, and we're still at the ground floor of figuring out what plants it works in and how well. But we are still learning what all there is to offer from it. There's a lot of questions we have. How can we make it better? Are there different ways we can do it? And so what we're going to talk about a lot is what we know so far, but there's still a lot that we have yet to learn. So to kind of give an overview of what we're dealing with here, go back to our example we had before. So one of the things that is used a lot in Cas9 is how do we turn a sequence off? So in this situation, my arrow got moved, but if you had the "did the nun eat the bun" sentence and you didn't want to say that anymore, what you could do with Cas9 is you can locate that sentence specifically and change it. So in this situation, then, we would be able to change the first letter, and so the first letter was a D, and now we've changed it to a G. And so now that sentence no longer makes any sense. And so that's how you would do it in the genomic sequence. You just find the sequence you want and alter it so it no longer would produce a product that you're interested in. Another option is to remove it entirely, so now that sentence is completely gone. And so you just basically cut it out, and so it's obviously not going to be functional anymore because you've removed it entirely. Like traditional transgenics, we can also worry a little bit about how would you insert a new sequence or how would you mutate one to make it say something different. So for instance, before we wanted to say the rat was fat for some reason. One of the ways that you can do that is you can replace a sequence that already exists. That's something that's new in the Cas9 system. So before, in our little paragraph at the top, it used to say the cat was fat up at the top, and now we wanted to say the rat was fat. So you can either replace that sequence entirely. And so to do that, you basically just locate the sequence that you're worried about, and then you can replace it. So then now we've got a new sentence that's now replaced our old one to say what we want. Okay, so that's one way to do it. Obviously, another option would be to find the sequence that you had before. And obviously, the cat was fat is very similar to the rat was fat, and we can just simply change the C to an R. So we just change one base pair or one letter in the entire genome, and it changes it over to what we want it to say. So that is something we had no ability to do before. We could never just find a sequence and change it to something different. This is very, very precise and something that we're able to do now with pretty good efficiency. So this is, and I know this looks like words and it's like the English language, but this is really kind of how it works with sequences. So we can just change a single base pair over. We can do all these manipulations in a genomic level. One of the things that gets talked about a lot is that these are basically causing mutations, especially with the turning it off and changing one base pair around. And so a lot of what it's causing are called SNPs, which are just single base pair changes. And all that means is that we're changing one base pair to something different. And that is exactly the same kind of thing that can happen in the natural system. So mistakes that will happen during DNA replication and repair, things like being outside in the sun, which plants are exposed to a lot, those are cause a lot of mutations, and so those base pairs will change on their own. And so the only difference is that we're causing them to happen in a direction that we choose, as opposed to having to wait for them to happen naturally. So we're kind of just accelerating a lot of the processes that would normally happen. And one of the things I think it's important to point out to people is just how specific this is. So to give you some context, so if you were looking at corn and you wanted to print out the whole DNA code, there's 4.6 billion characters in it. And so if you were to print that out into a book, it would be equivalent of 14,000 Bibles thick. You've all probably seen the Bible, so that's how thick it would be. Okay, so it's a huge book. And so if we were doing traditional genome engineering, it would be equivalent to basically taking that entire book, flipping through it, and throwing a sentence inside of it. And so then you'd have to go back, see where the sentence was, make sure it's not interfering with anything. With traditional genome engineering, it's basically like putting them into a word processor and then basically doing a search and then replace. And that's exactly how precise it would be. We know exactly what we're changing. We know exactly what's going to happen when it's done. And so the specificity is really what's allowed us to be so excited about it. And so as a summary, it's pretty easy, especially from a technical perspective, which I didn't really touch on. The idea of doing it is not all that difficult, especially compared to what we were doing before. It's specific. But in contrast, the thing that's hard to realize is that you're also still reliant on the target genome. And so one of the things in the lab that I mentioned that we're working on is how do you introduce new genes? How do you work with what you're given, which is something we didn't have to work with before. So I hope that kind of helps explain the advantages of how CRISPR-Cas9 work. So to move on to the next question, which a lot of people I'm sure are very interested in, is why is it that a farmer would care about it? It's obviously a very academic thing. Why should a farmer be interested? And it's really an interesting question because why should anyone be excited about it? And so the biggest takeaway, obviously, is the specificity. It allows us to pick exactly the sequence we want to mess with and change it exactly how we want it to do. We can introduce natural changes, and you can also do this without including that machinery if you use the tricks correctly. these last 3 points all go together about regulation. If we're introducing a mutation that can happen in nature, and we don't introduce anything new to the genome that we're messing with, okay, so then we've got a sequence that is exactly the same that could occur in nature. That goes together with regulation. So is it possible to then have less regulation on the products that come out of it? You know, traditional genetics are going to allow you to do a lot of cool things, but it also takes tens of millions of dollars in like a decade to get them approved for release. So if I, as a researcher, come up with a new technology that allows a corn plant to produce a new defense kind of a system, then we're not able to release that into the public because we don't have tens of millions of dollars to go through the regulatory system. And so it's basically cost prohibitive unless you're a big company. Gene editing is going to allow us, hopefully, to not have to go through such a regulatory process. So it's possible that the smaller companies and researchers in the academic labs might be able to have a say in the matter. And so we might be able to have crops that maybe don't have as high of a return able to be released for growth and into the public sphere because it's less of a cost of release. So really, gene editing is kind of the future that we see of how agriculture is going to go forward, especially from a manipulation standpoint, where the lower input costs and regulatory costs is going to allow us lower barrier to entry. So hopefully, we're able to then see more products come to market. And so all of this kind of comes together with a less of a research time. It's all about less cost, less regulation, meaning that hopefully smaller business and academics like myself can actually get into the game a little bit to where the things that we're able to make successes with, we can hopefully bring out into the market. And so then what are the latest advancements made? And so this is more of a question for Michiel, where from a business perspective, I obviously am not a company person. But I can tell you that the advancements in Cas9, the whole CRISPR technology, are pretty much every plant that I've seen has an ability to be modified with the technology. But as I said, the technology is very new, and so products are just starting to hit the market. And so we're hopefully there's going to be a greater diversity, more crops. Maybe the crops with a little bit smaller of a margin are able to be commercialized and have a bit of a profit on both ends. And so one of the things in my lab is that my boss, Dan Voytas, he started a company when these technologies first started hitting the market because he saw the potential of it. And so I don't know much about the company, but I pulled from their website. This is just a list of, straight from their website, of the types of products that are coming out of just a small company in Minnesota. So you can see there's two things where you can have consumer-centric products, things that are pretty much consumer traits as opposed to just herbicide and insect resistance. Things like a lower browning type of potato, a different oil composition in soybeans and oil products, in addition to like the herbicide tolerance and mildew-resistant wheats and things. And so these are all coming down the line, and they've already seen some of them are allowed to not have as much of a regulated article in USDA, which means a lower cost for entry. And so these are the kind of things that really excite us as academics and researchers, because these are smaller companies that are able to finally get out to the public and release kind of the things we've been working on for the last few decades. Maybe we can turn them in with this kind of a technology as a benefit to the consumer. And so then where will the future of plant breeding go? And so this is a question that's kind of interesting because there's no plant breeding. I kind of consider plant breeding and gene editing kind of as separate but the same. And so a lot of what plant breeders do is you take a giant field and then you kind of look and see which plants are going a little bit better and use those as your germplasm going forward. And so you always have a continual improvement. That will probably continue to be the case, but we also are able to take the germplasm that's produced with that and then use the Cas9 technology on that improved germplasm. Additionally, as breeders identify different traits, we're able to modify them with the Cas9 technology as well, so maybe we can have an accelerated breeding program in using these kind of technologies. A lot of breeding relies on what we call quantitative traits. This means it's not just one sequence that contributes to them, it could be hundreds of sequences that contribute to a given trait. And so obviously from a Cas9 perspective, you would have to modify each one of those traits individually or each one of those sequences individually, which is not feasible at the time. But we can take the improvements we're able to make with Cas9, apply them to the breeding perspectives we have now, and kind of accelerate them. So it's always important. Everyone always says, whenever they give any of these kind of presentations, that this is just another tool in the toolbox. Everyone says it. It's very true. There's no reason that what we're doing now with CRISPR-Cas9 is going to necessarily mean that we're going to stop doing breeding programs, or we're going to stop doing traditional transgenics even. These are all just improvements that we think that all going to come together to better improve what we're working with. And so with that, I will stop talking at you and let Michiel take over next, and I'll be happy to take questions at the end.
Thank you so much for that, Nat. I loved hearing you talk about how different you could look at CRISPR-Cas9 and how it was just like looking at words in a sentence. If you have any questions, please feel free to go ahead and type those in the chat box now, and I will address them for you during our Q&A session. Until then, we will move on, and our next speaker is Michiel van Lookeren Campagne. And Michiel is the global head of seed research at Syngenta. He's based in Research Triangle Park, North Carolina, which is home to Syngenta's Innovation Center and Advanced Crop Lab, a research facility that he was instrumental in founding. Michiel received his university education and doctorate in biology from Leiden University in the Netherlands, and then he spent 5 years in New York, starting as a postdoc and ending as an assistant professor at Columbia University, where he worked on signal transduction and cancer. Michiel entered the ag industry in 1993 and joined Syngenta in 2009. During his tenure at Syngenta, he has been an integral force behind the company's adoption of cutting-edge plant breeding innovations. Michiel, thanks for being with us here today, and I'll let you take it from here.
Yeah, thank you very much. And of course, it's always a pleasure to be able to talk about science and products coming from science to growers. What I will try to do is trying to let you sort of see how we are thinking of genome editing as the next step, or the next tool that we will use in plant breeding. If you look at plant breeding, started of course with crop domestication about 10,000 years ago. And then, in 1865, Georg Mendel, with his famous pea experiments, really made us understand genetics, how that works, and inheritance. And that then led to, in the 30s, the first generation of corn hybrids, first with a triple cross and the double cross, and then, when in 1953, Watson and Crick described the structure of DNA, that entered then the era of biotechnology, with the first products of GM reaching the market in 1994. And also the whole marker technology, which is now an integral part of any industry doing plant breeding. It's really, really now in every crop we use that, and it is enormously beneficial tool for our whole plant breeding concept. Now, of course, what is then the next thing at the horizon is this genome editing thing, or this CRISPR. CRISPR is one tool of the genome editing. In fact, we've been able to genome edit crops already since 10 years, but the process was very, very cumbersome, very, very expensive. And the invention or discovery of CRISPR-Cas9 really has lowered the cost enormously, made it accessible to any self-respecting molecular biologist can now edit a plant. So then you see what all this whole modern plant breeding has done in the last decade. So since 1935, we had the first double crosses and these single crosses, and then the GM CRISPR. That has really been, all these new technologies have been able to keep us improve, in this case, U.S. corn yield by about 1% on average a year. We have been consistently being able to improve that, because every time we were able to deploy a new set of technologies to keep that up. And that improvement was 40% due to agronomic practice, but 60% really due to genetics. And that is driven by the breeder's formula, where we call the genetic gain, which is driven by the genetic variation, the amount of selection pressure you can do on that, and the accuracy with which you can measure it, and the time. So those are the 4 parameters that we're always trying to work on in plant genetics. Can we increase the genetic variation? Can we shorten the time? Can we more accurately measure going forward? So in fact, genome editing is a tool that can help us in this genetic variation, but also in the time. I just wanted to sort of compare these 3. So you have your conventional breeding, you make a cross, and what you basically do is you reshuffle all the genes from the 2 parents, and then you optimize the performance. So you really have all the genes in play. If we take corn here, that is between, it depends on the crop, between 10,000 to 300,000 genes, and that process then takes about 7 years to then develop a new variety. And it is really limited by the range of traits and what you have in the germplasm that you use for that breeding program. With genetic modification, we can do something else. We can take a gene that we have well studied, say it's like a BT gene that converts insect resistance, and we can take that from a bacteria, put it in the plant, and achieve that, a trait for that. That takes us about 10 to 12 years and costs us about $130 million to develop. So you see that is a very long time, and it is high cost for product development, and that's made it really restricted that technology to really a couple of really high value traits. And if you look at it, basically we've got insect control and herbicide tolerance as the 2 major traits that are there. There's a couple of other ones, like our Enogen corn, where we use an enzyme to make starch easier degradable for the ethanol industry, but those are really rare exceptions out there in the market. And then if you look now what we can do with genome editing, we can precisely, as Nat already told us, edit a gene that is in the crop and then achieve the desirable trait. And we can do that actually much faster. So because we don't have to make all these crosses anymore, we can do that in 3 to 5 years instead of the 7 years. So the ideal would be then is that if you have a parent line that you're going to use for a hybrid, that you make the edit in the parent line, and then you use that improved parent line then as a crossing partner for you make your next hybrid. So that can really speed things up. But of course for that, we need to be able to edit commercial germplasm, and that is really one of the things we're really working very, very hard on to make sure that we can edit the elite lines, so we can do all these conversions in parallel. We can edit these lines in parallel and get the product out to grow as much faster. But the other thing that we sort of struggle with is knowing what to edit. As Nat said already, most of the edits that we see now coming to the market, the first generation of traits coming through that, are really knockouts of genes in a crop. Now you can think of, if we take those 14,000 or 140,000 Bibles, if we take out one page, you're not going to improve that. The chance that you improve that text is not going to be very good. What we really want to take out is the spelling mistakes, and perhaps improve some legibility or change it to modern spelling if you want. So for that, we really need to know what to edit, and the biological knowledge that we have is really quite limited still. So we think that the real benefit from genome editing will come over time, but it's going to take another 10 or 20 years before we have the full benefits of the technology. There will be some low-hanging fruit coming here now, but the real benefit will be an integral part of the breeding program as we know that. So in summary, it is as Nat also said, it's a new tool in the toolbox that allows us to keep that improvement going year on year and get better germplasm, better varieties out to you so that you can plant them in your fields and have higher yields and really protect your profitability as a grower on your farm. It's a more efficient way of developing seed because it reduces the time to development by about 2 years. And of course, it complements all our other tools like breeding, like GM, And so in a nutshell, the CRISPR, it edits the text, and the genetic modification, adds a new chapter. But all of these tools are very, very important for us to develop new varieties and get them out to you as fast as we can. What are then the benefits for the farmer and the value chain? What it allows us to do is to accelerate the rate of innovation in the development of new plant varieties, bringing novel traits into your hands with greater efficiency. So, as Nat already showed you in that list of calyx traits, these are improved grower traits such as improved disease resistance, improved drought tolerance, higher yields, but also consumer benefits such as better food quality and nutrition and longer shelf life. All of those traits are addressable with genome editing. Just as an example, we're working on many traits here. But as an example, we're working on drought tolerance for corn, and we've edited a gene that we found from our discovery program and made a designer change. So we changed, in fact, one amino acid, one of these letters in that presentation, to then create a more optimal version of that gene. And that then used in our greenhouse test where we have that now, we showed that those plants can perform with less water. And of course, that's in improving on our technology. So like we have our artesian hybrids that are water optimized hybrids. This is in the next generation pipeline. These type of traits will come into that in that artesian type of product to the market and to you. And of course, we are not quite there because the genome editing really started to take off just a couple of years ago. So it'll take a couple of years for us as well to bring that to the market. And our portfolio there will come to the market in the beginning to the mid of the next decade. And you see that we can, we're using this in multiple crops. At the moment, we have active programs in corn, in soybean, in wheat, in tomato, in rice, and in sunflower. So all those crops we're able to edit at this moment, and we have active pipeline of projects trying to improve those crops to get them out to the growers as fast as possible. You can see some of the products that are already out there, not always done by CRISPR, but by other editing technologies that the U.S. has deemed do not have to be regulated. For example, with a different type of editing technology, Voytas has introduced herbicide tolerance in 2015. The mushroom anti-browning trait was actually made with CRISPR, and it's the first CRISPR trait that the USDA has publicly stated that does not need regulation. And then there's other things like wheat powdery mildew resistance, high oleic acid oil, waxy corn, and disease-resistant rice, all products that are reaching now the market where we basically use this high tech to make a alteration in the gene in a similar way that we could have done by breeding, but much more precise and much faster. So this is all reaching the market quite fast. Now, the U.S. is, of course, always at the front of these type of innovations, and that's one of the reasons that I work in the United States, being from Europe originally, is because of the technology acceptance and the appetite of growers for high tech. But of course, we as big companies need to be able to transport our crops around the world, develop them for other territories as well. And for example, in Europe, we do not know yet how the regulatory agencies are going to treat this kind of innovation. So we are working hard to make sure that we get a globally harmonious path to the market for this, and that we do not get into the trap where we reached with the GM technology, where we are stuck with very, very high costs and very, very difficult regulation that is different everywhere along the globe. And show you a little bit the importance of that. Because here on this graph, I show you that, for example, in the red, if you have in the red line, you see that if something costs a lot, the technology adoption rate can only be high in crops where there is a high gross profit pool where you can amortize those investments again. And that's why you see why we have only GM traits in corn and in soybean, because those are large crops. Together, corn and soybean have 60% of the value of the total seed industry is in corn and soy, and that's why corn and soy can afford GM crops. But other ones, like in the medium sector, they're like sunflower, or perhaps a smaller crop like lettuce, will never have access to GM technology just because it would not be affordable, or perhaps because they are grown in areas where the technology is not accepted. If you then look on the other side on the green line, is if you say like marker technology has become low cost. We can now deploy them widely. It used to be first in corn, and we had a very, deployed them in corn and soybean. Then we went to sunflower, and now we are deploying markers, genetic markers, in all of our crops in Syngenta. All of the crops that we bring to growers, we have these marker technology. And then, of course, you have these medium cost ones that are perhaps in between, like genome editing at this moment. So if we can sort of migrate this at medium cost, what we have now, the genome editing, and migrate it like the markers in the low cost, then, of course, the amount of innovation that we can bring to growers is just going to be much broader and much more profound and faster than what we've been able to do with the GM technology. There's so many in my history in the industry that we have just had to stop so many very, very interesting grower-centric projects in the GM area because there was just no return on investment or there was no acceptance for them, that it is really heartbreaking. And we just hope as scientists and as industrial scientists that the regulation here will allow us to develop this technology to its full potential, to bring innovation into the growers' hands as fast as possible. Thank you for listening to us. Additionally, of course, you can find information about Syngenta on these websites, and of course, you can also connect to us on social media as well. And with that, I'm finished and hand it back to the moderator. Thank you.
Thank you so much, Michiel. So we're now going to move right into our Q&A portion of the webinar, where we will answer any questions you might have had throughout the presentation. As we move into this session, I encourage you to type any questions you have into the chat box. Because this webinar is being recorded, our first question is, can I get a copy of both presentations? Because this webinar is being recorded, we will post this online within the next 48 hours. So you will be able to watch this again afterwards. So what makes the timeline for CRISPR products so different from GM products? And how about we start with Michiel, since you spoke the most recently, and then we can jump over to Nat to see if he has any extra thoughts.
Yes, so in principle, the timeline could be similar. No, so there's no reason why GM could not be fast, because you can insert a GM trait. So it's an insertion of a piece of DNA that encodes for, say, insect control. You could insert that into the genome, and if you could do that into an elite line, you could do that in one go, and you would bring that to the market. But because we have we have all these controls and safety measures on it, we can't do that because every insertion point then would be different, as Matt already said, because it would be at random in all these different elite lines. And we then, of course, would have to test on each of these lines, do they work? And that are they, is it express? Is there a safety issue? We go through all the regulatory process, because we have to do that in all these countries. And it is really the regulatory process that adds these timelines on the GM event versus the genome-edited event. If you would have the same kind of regulatory controls over a genome-edited event than over the GM, then also, of course, the genome editing would not have any advantage whatsoever. The idea is, of course, now is that what you do with genome editing, you let the plant create its own new mutation. That's in fact what you do, because you let it repair a break that you do in the plant. So it is no different than any other mutation that you would do as described by Nat previously. And so there's no foreign DNA coming in. So it should not be under the same regulation, and it should just be seen as a breeding tool. And I think that's the argument we are always trying to make, but it's of course up to the regulators to decide how it's going to be regulated. So I think that, in short, the timeline difference is really due to the regulatory process.
Right. Well, thank you. Nat, did you have anything you wanted to add to that?
Yeah, I mean, that's pretty much it. It's interesting because there's basically, the way I look at it is two parts of this, right? There's producing the gene-edited plant, and there's taking that produced plant and moving it to market. And the process of producing the gene-edited plant really shouldn't be all that different from a traditional transgenic. There's probably, depending on how much resources you have, there's probably more screening, and so there's a more labor cost for making sure that it's, the product is what you want it to be. But if you're a company, like a big company like Syngenta, for instance, then you have the resources to kind of do that all at once, and so the overall cost may not be that different. But getting it to market, that's a completely thing I'm not all that familiar with, but if you have to go through 15 years of regulatory for one, and you only have to go through a couple of years of regulatory for the other, then there's a huge difference in time there.
Okay, well, thank you, both of you. Our next question comes from Robert, and he asks, how can you test whether a product has undergone genome editing? I'm going to throw this to you first, Nat, and then we'll see if Michiel has some additional thoughts afterwards.
Yes, so that's kind of an interesting question. So it depends on what you have done. So there's the technical aspect of, are you only changing a single base pair? Are you changing one letter in the genome? If that is the case, then you probably can't screen for whether it's undergone genome editing or not. And that's pretty much because it's basically simulating a natural process. So if you're doing the exact same results as what you could do just by a breeding technique, you're probably, the end result should be exactly the same. And so it's not really easy to find whether it's been gone through a genome editing process or not. On the other hand, if you're introducing a new sequence or you're removing a whole sequence, for instance, you can go through the genome and look and see those changes. But, it's really going to depend on what the product is, what it is that you're trying to do. And that's kind of the big question in the regulatory sphere that we keep hearing is, are you editing by just changing a base pair? Are you removing a whole thing? Are you adding a whole new sequence to it? And those different changes will probably change what you have to do to get it approved from market.
Yeah, so that's my side. What we do, of course, when we make an edit, is then we always check whether the edit that we made is the intended edit, and we do that by DNA sequencing. So it's very, very simple. We take the plant that we think has been edited, and then we sequence that area, and we see, okay, the intended change has been made. Now, the tricky part, of course, is if you want to then regulate this, the problem is that regulation often is done by process. So, and you can't see from the plant how it has been made. It is, so it's going to be different. Like in GM, you can test whether it is an insertion there, and people usually use similar kind of genetic elements in there. So you can do quite easily, if you have a seed, you can see is it GM or is it not GM. In genome editing, the end product, you can't distinguish on how it was made. So that's going to be an interesting regulatory question, is how do you regulate a process versus a product? And of course, we've been we've been trying to talk to regulators that it makes much more sense to regulate the product rather than the process. So in fact, you could not see how it was made. I always say it's like you can't see from a car that you're driving whether it was made by people or whether it was made by a robot. You have to ask the car manufacturer. So this is the same here a little bit. Is you cannot test a genome edited batch of seeds whether it was genome edited or it was a mutation that we found naturally occurring. That's a dilemma for the regulators at this moment.
Okay, great. Thank you for that insight. Our next question comes from Julie, and she asked, this is a long question, so if you need me to repeat it, go ahead and tell me. We often hear conflicting statistics of whether GM plants require overall more or less crop protectants. Do you think that some of these new plant breeding innovations allow us to reduce our reliance on hard chemistries? And I'll throw this to Michiel first.
Yeah, so it's an interesting question because it comes a little bit back to my former argument, no? So GM is a process. It's a way that we add genes to a plant. And if you, so obviously, if you're going to put, for example, a Bt gene in there, and it protects that plant against an insect, you will use less insecticides, and that has been widely reported and widely used. So there is a real benefit of having to use for the growers also to use less insecticides on a Bt crop. It's clear we don't have to spray, or perhaps there's some secondary pests or so. Really clear benefit. Now it's a little bit different when you look at the GM, say, of herbicides. So if you look at, for example, a glyphosate-tolerant herbicide-tolerant corn, obviously you're going to spray glyphosate on that to get rid of your weeds. Now, the question is then, are you spraying more glyphosate? Obviously, you're spraying more glyphosate because you would not spray glyphosate on the crop because you would kill the crop if it would not be tolerant. But you will probably not spray some other herbicide. So it's that is the question then, is yes, has there been more glyphosate sprayed in the world because we have glyphosate tolerance? Yes. But the overall pounds of herbicides sprayed on fields by using herbicide tolerance technology has clearly gone down. And there's now many studies that overall, and there's just recently came out an Italian study that was a meta-analysis, all the literature, that GM technology increases yield, decreases mycotoxins, and uses less agricultural inputs such as herbicides and insecticides.
Okay, great. Nat, did you have anything you would like to add on to that?
Not really. That's a brilliant answer. I would encourage anyone who's interested to go and look up that Italian study he's talking about. It really breaks down the benefits of GM technology over the last few decades.
Okay, well, thank you so much. I'd like to go ahead and thank our speakers for coming and taking some time out of their day to work on this webinar with us. Nat and Michiel, just thank you for sharing your expertise with everyone and for helping us go through this question of how CRISPR will affect our farm in the future. I know it's a bit of a big question to answer, and it doesn't have necessarily a lot of hard answers right now. So I'd like to thank you both for taking the time to come speak with us. And I'd also like to thank everyone who came and participated in our first webinar. I hope that you found this information of value, and we look forward to our next webinar. I'd like to also quickly thank my team working behind the scenes for all of their hard work in making this a successful event. Again, as we mentioned before, this presentation will be made available online at american-seed.com within the next 48 hours. Thanks again, and we hope you all have a terrific day. This is Alex Martin signing off.